Hello friends, let's go through this introduction to carbohydrate metabolism. So what is a carbohydrate? It has two forms in nature, D or L. The D form is found in the human body, and there's also the alpha and beta form. Beta is where the CH2OH group is going to be above the plane in the D-glucose, and it's going to be below the plane in L glucose or any kind of carbohydrate. The example is just glucose, but even if it were like a D-lactose, the CH2O2 group would be above the plane. So all of the polysaccharides and whatnot are held together by the glycosidic linkages. We'll go through that in the next couple of slides. And the keto alcohol is the reducing end of the carbohydrate. The functions of the carbohydrate is a biological fuel, the precursor of important substances. It plays a role in the structural integrity of the cells. It also is a part of the cellular membrane in order to create the semi-permeable membrane. It also plays a role in like signaling, such as like being part of the immunogen. It's also storage of water. It plays a role in cell adhesion as a cellular cement, and it's also a part of the anticoagulants. Carbohydrates can be monosaccharides. Here are a couple examples, and they are furans, which are 5-membered rings, and pyran, which is a 6-membered ring. These are the only ones that exist in great amounts, because any other numbered ring below the 5-carbon ring, so like a 4-carbon ring, 3-carbon ring, those are too unstable for the body to use. The 6-membered rings are actually the most stable molecules, so it's rare to see anything above 7, 8, 9, 10. Like those do exist in the body, but this is the more common one because of the amount of linkages it's able to create. Aldopentoses and ketohexoses are furanosic, so these will form the 5-membered ring, while aldohexoses and ketoheptoses are puranosic, so those will form the 6-membered ring. Aldoses are the most oxidized because those are aldehydes. So polysaccharides, amylopectin has branches every 24 to 30 subunits, so every 24 to 30 of this is going to branch off like that. The subunits are usually maltose and isomaltose, while the amylose subunit is maltose, which is unbranched, and it's helicoidal, so it will be in a helix. These play an important role in all of these molecules, but most importantly, it plays a role in the nucleoside synthesis, and it's a reduction derivative in metabolism. Disaccharides are going to be 2 sugar molecules joined together. These are the most common ones. Maltose is going to be 2 glucoses at a alpha-1,4 glycosidic bond. Sucrose is 1 glucose and 1 fructose at a alpha-beta 1,2 bond. So this is sucrose right here. So this is the 1 bond, and this is going to be join beta, so beta, remember, is above the plane, and this is joined at like carbon number 2. And isomaltose is a glucose along with another glucose, but this is at a alpha-1,6 bond versus the alpha-1,4 bond. And lactose is going to be a galactose along with a glucose at a alpha-1,6 glycosidic bond. Polysaccharides are macromolecules. They are They can be homopolysaccharides, which is like a long chain of the same subunit, or heteropolysaccharides, which is a long chain of separate and different subunits. These are examples of polysaccharides. Glycosaminoglycans are repeating disaccharides, and it's composed of an amino sugar and an acid sugar. So here is hyaluronic acid, the most abundant glycosaminoglycans in the body, and here is the amino group, and here is the acid sugar. Some of them can be sulfated, and they exist on the cellular surface along with the extracellular matrix. These are other examples, and all of them are like proteoglycan linked. Glycogen, unlike the other units, molecule has 8 to 12 subunit branches, and it's made out of maltose and isomaltose. The branches give us the non-reducing end, so that can create a lot of glucose. So like every time it splits at the non-reducing end, it frees up a sugar molecule. Glycogen lowers the osmotic pressure because you're able to pack so much potential energy in a relatively small molecule using the glycosidic linkage. And they are predominantly in the skeletal and muscle and the liver. And the liver glycogen can be used everywhere once made, but the muscle glycogen is selfish and it's only made for the muscle. These play a role in like the structure of the muscle, the regulation of water metabolism. It's also it plays a role in adhesion. Once again, it's a biological sieve, and it plays a role in lubrication because carbohydrates are hydrophobic, and it also is a docking site for growth factors. And growth factors are, of course, they play a role in like healing. So they can be proteoglycans, which are proteins containing covalently linked glycosaminoglycan chains. Alright, so let's go through metabolism really fast. The basic gist of metabolism is the release of energy and the storage of biological fuel. In the synthesis of glycogen, it is going to create energy in the form of NADPH. And the sources are, of course, starch, sucrose, and lactose. And the most important carbohydrates are going to be actually starch, lactose, and glucose. I'm not sure why I put sucrose there, but it should be glucose. Alright, saccharidases will hydrolyze the linkage, glycosidic linkage at the alpha-1,6 branch point, and amylodextrin is a starch fragment that stains blue. So, it's an important clinical indicator. So this is the general overview of, polysaccharides all the way to glycogen synthesis. So starch is going to be broken down by amylase that is in the mouth and in the stomach. And it's an endohydrolase, so it will break down the starch within, not just at the ends, and it will break down into either these 3 molecules, and then from there it will have certain specific endohydrolases, and that can come from the esophageal and pancreatic cells, and then the disaccaridases will metabolize these structures into monosaccharides, and then that will create, remember, sucrose makes like 1 glucose, 1 fructose, maltose makes 2 glucoses, lactose makes 1 glucose and 1 lactose, and those can be further degraded until we form glycogen. So the digestion of starch begins with the cleaving of the alpha-1,4 linkages until it reaches a alpha-1,6 linkage, and then it can't use the original enzyme that cleaves the 1,4 linkage, which is the glycogen phosphorylase. So once it reaches this branch, glycogen phosphorylase stops, and then the branching and debranching enzymes will cleave the alpha-1,6, and then there's going to be a transfer. So like this new little like a freed branch, like the alpha-1,4 will cleave until here. This will move and attach down to here using some kind of transferase, and then it will continue cleaving, each making an individual glucose, and then this will cleave, freeing this as a free glucose. Okay, so it's going to be absorbed via a secondary active transport in the intestines. SGLT1 is a co-transporter, and the energy of the for transportation will come from the electrochemical gradient. GLUT2 is an important transporter because GLUT2 is a primary role of carbohydrate metabolism in the pancreas and the brain, along with the liver. These will sense the amount of glucose in the body. Fructose is transported via the facilitated diffusion through some kind of like protein pore. And GLUT5 is going to be in the apical membrane of the intestines, and GLUT2 is going to be in the basolateral membrane. So GLUT5 is going to be in the intestines, while GLUT2 is going to be in primarily like the brain, pancreas, liver. Okay, here we go. So GLUT1 is in most tissue. It's going to be a basic basal uptake. It's how we transport carbohydrates through the blood-brain barrier. GLUT2, once again, is mostly the kidney, liver, and pancreas, serves a role as a glucose sensor. GLUT3 is going to be mostly in the neurons and the placenta. This is a high affinity transporter. Makes sense. These two are very demanding. We need it in the brain to provide energy, and we need it in the placenta to provide energy for the developing fetus. GLUT4 is going to be mostly in skeletal muscle and adipose tissue. It is an insulin-stimulated glucose uptake, and it's stimulated by exercise, of course. GLUT5 is going to be in the intestines, and it plays a role in fructose transport. GLUT7 is in the gluconeogenic tissue, which is basically just the liver, and it mediates the glucose flux coming in and out. Okay, so these are the important differences between hexokinases and glucokinases. So hexokinases are in all tissues, while glucokinase is only in the liver. This is an allosteric enzyme. This is not an allosteric enzyme, and this is insulin dependent, while this can have a variety of other limiting and regulatory factors. And hexokinase has a broad specificity, while glucokinase is very specific. Okay, so the GP6P formed from the catabolism of carbohydrates can go into glycogen for glycogen synthesis. It can also be a part of gluconeogenesis or glycolysis, and it can go into the Krebs cycle as pyruvate, or it can go into like the synthesis of nucleotides via our ribose 5-phosphate. So, of course, with metabolisms, there can be some problems, and they're generally hereditary defects, which are the deficiencies of individual disaccharides or intolerance. Breast border enzymes are lost during the severe diarrhea, and that can cause temporary intolerance. And, the generalized defects, these are the generalized like symptoms, so intestinal diseases or malnutrition, drugs, intestinal injuries, so a colonoscopy gone wrong, or an excision of some of the bowel gone wrong. Lactose intolerance are found mostly in Asians and African populations, while isomaltase sucrase is far more rare, and it's usually found in just the Inuit or the Eskimo population. Alright, thank you for your attention and good luck studying.